Right, welcome back to our third lesson on respiration, and this is all about oxidative phosphorylation, which is one of my favourites because it is, I think, really quite cool. And hopefully it won't be as intimidating as the light-dependent reaction was in photosynthesis, because a lot of those principles were brand new. We're actually going to revisit a lot of that, and a lot of this should hopefully look somewhat familiar, because oxidative phosphorylation and the light-dependent reaction have a lot in common. There's like chemiosmosis, we've got an electron transport chain, we've got ATP synthase, all that. So there's a lot more in common. There is obviously some deviation because it is a slightly different process, but hopefully a lot of the machinery will be familiar to you. So what I've created is a series of PowerPoint diagrams, which we're going to cut to in a second, to help break this process down bit by bit, so you hopefully get a good understanding of it. So the first thing that happens is all of that reduced NAD and reduced FAD that we've made in the glycolysis, link reaction and Krebs cycles is all going to end up here. It's all going to end up in this process, which occurs in the inner membrane of our mitochondria. Embedded within that inner membrane is an electron transport chain, so we have a series of protein-based electron acceptors. We're going to have ATP synthase sticking through, and then between that inner and outer membrane is what we call the intermembrane space, where a lot of this process will work with as well. So all of that reduced NAD and FAD has arrived. It's now here at the inner mitochondrial membrane. And those hydrogen carriers, those coenzymes, will release their hydrogen atoms, and they will immediately be split into protons, H+ ions, and electrons. Okay, so the electrons will immediately be accepted onto the electron transport chain, and they'll be passed along in a series of redox reactions, and they'll be transferring energy as they go, going down in sort of progressively lower energy states as they go. One way I like to think about this, and I should have really talked about this when we did photosynthesis. Imagine I got a hot baked potato out of the oven, high energy, hot, and I chucked it to you. You'd grab it and it'd heat your hands up, wouldn't it? Hot, hot, hot. You then quickly pass that on to the next person. That'd be a little bit cooler now because it's transferred some energy in the process. And as you passed it along from person to person, the potato would cool down, wouldn't it? Because it's been transferred to your hands in the chain. And that's a silly way of kind of thinking about this. So hopefully that helps. So those electrons are passed along and they transfer energy. What's that energy used for? Well, that is the next stage. So those protons that we have also produced, they're going to now be actively transported from the matrix into that intermembrane space via those electron transport chain proteins, because they also act as pumps. So they're going to actively transport those protons into the intermembrane space. So now we're going to build up a very, very high concentration of those H+ ions in that intermembrane space, and of course we're going to have a lower concentration in the matrix because we're constantly pumping them out. Now, the structure of the mitochondria is important. So those membranes that make up the inner and outer membrane are impermeable to H+ ions. They're charged, they can't get across. So they can only get in via the ETC proteins, the electron transport chain proteins, and they can't get out. They're stuck. So we build up this very, very large concentration of them, very high concentration. The only way they can get out, and hopefully you already figured this out, is through ATP synthase. So we've got this steep concentration gradient, and they're going to go down it, and therefore through that ATP synthase. That flow, that proton motive force as it's called, will actually cause the shape of our headpiece of the ATP synthase to change shape. And that change of shape will provide the energy and the force to combine ADP and phosphate into that energy currency molecule ATP. And this is where we generate loads and loads of that ATP. Top of my head, I think it's about 34 molecules of ATP are produced in this way per glucose molecule. Another way of looking at it is to think of chemiosmosis theory. It's a bit like how a hydroelectric dam works. So how does a hydroelectric dam work? Hopefully you know this from year 9. We build a dam, we build a wall, and we trap flowing water behind it. All that flowing water has gravitational potential energy stored in it. As it's flowing, it wants to flow downhill, it wants to transfer that energy. So it's a huge store of energy. The dam prevents the release of that energy, and the only way it can get through is through turbines. So as that water is flowing through, it turns to turbines, generates electricity. And this is what's going on with respiration, isn't it? Instead of water, we've got these protons. They've got potential energy in them, okay? And they want to flow out, and the only way they can is through basically these biological turbines, the ATP synthase molecules. It's really quite remarkable how something that is literally billions of years old, like ATP synthase, works incredibly similar to our modern electric turbines. Really quite extraordinary how nature, well, I should say human industry is kind of imitated nature without even knowing it until much later, because we've had turbines longer than we've known about these enzymes. So that's it. We generate loads of ATP, and then there's one last little bit that ties this all off. And this is where oxygen comes in. So we know, hopefully you have an idea that all these processes, the link reaction, the Krebs cycle, and now oxidative phosphorylation, are aerobic. They require oxygen. The link reaction and Krebs cycle didn't require oxygen specifically. They needed it to be present in order for them to operate. Here we're actually going to see where oxygen is used. And oxygen acts as what we call the terminal electron acceptor. And it sits on that very last protein within the electron transport chain. And as those electrons are passed along, going down to lower energy level, they finally are accepted there by the oxygen. And those outgoing H+ ions will combine with that electron and that oxygen to make water. Okay? And that brings us full circle on our equation. And I'm going to jot that out on the board in a second, and we'll have a look at that and talk about it to summarise what we've seen so far. And that's very important that you refer to oxygen as the terminal electron acceptor. What would happen if there was no oxygen there? Well, we'd have this big knock-on effect. If we didn't have oxygen, either because our cells become anaerobic or because something like cyanide, which actually acts as an inhibitor, it blocks, it prevents the oxygen, it binds in place of oxygen. What would happen is that we would prevent the oxygen being the terminal electron acceptor. As a result, the electron transport chain would stop because it wouldn't be able to accept more electrons because they're not leaving the system. As a result, we wouldn't be able to oxidize our NAD and FAD. They wouldn't be able to release their hydrogen. And as a result, they wouldn't be able to oxidize those intermediate products in the link reaction and the Krebs cycle. Everything would come to a halt. Okay? So that's why oxygen is very, very key there. So let's look at now then at that equation to summarise everything we know so far about aerobic respiration and where all those different bits fit in. Cool, after all that lot, I need a cheeky cup of tea, I reckon. Beautiful. Right, okay then. So let's summarise everything that we've looked at so far when it comes to aerobic respiration. So the glucose came in with glycolysis, didn't it? I'll just put a G there for that. So that was right at the start of the process. That happened inside the cytoplasm. The oxygen we saw didn't come in until right at the end of the process in oxidative phosphorylation. So I'll just call that OP there. The carbon dioxide we saw was released in both the link reaction and the Krebs cycle. They both were the source of our CO2. And then the water was that final product of the oxygen combining with the electrons and the protons. So we saw that again in our oxidative phosphorylation. So we can see that how all four processes link into our nice simple word equation that you learnt all the way back in year 9. Now we understand how it all works. Okay. There's two little last bits we can talk to, and again I'll cut to a diagram for each of these to help you get an idea. And that is the use of other respiratory substrates besides glucose. So how can lipids and how can proteins be used to help synthesize ATP? Okay, so we'll start off by looking at lipid metabolism. So now I've got this diagram up on the screen. We'll have a look at it and break it down. So the first thing that happens is the lipids are hydrolyzed into glycerol and fatty acids, and they're treated differently in order to help generate ATP. So the glycerol can be converted into TP, and we saw that TP was one of those intermediate products in glycolysis. So basically it can plug straight into glycolysis there, those glycerol molecules. The fatty acid chains have to be further hydrolyzed down into 2 carbon chunks with their hydrogen attached. And they effectively can act in the same way that acetate does, and they will bind with acetyl-CoA, and therefore they can enter the Krebs cycle. The thing about lipids is they actually contain significantly more hydrogen atoms than carbohydrates do. And as a result, they can actually reduce more NAD and more FAD. And therefore, they can provide us with more potential energy, more ATP synthesized. Another added benefit of lipid metabolism is that because it's got more hydrogen, there's more hydrogen to combine with oxygen. And therefore we can generate even more water, metabolic water, as a result. And some animals, like something like kangaroo rats, jaboas, whatever they're called, can actually go their entire life without drinking any water at all. They can get all of their water they need metabolically through using by using lipids as a respiratory substrate, because they eat seeds which are rich in lipids. And actually that way they can generate all of their water they need. Really quite remarkable. The reason why we perhaps ourselves will preferentially use glucose over lipids is a little bit more tricky. I think it's because the glucose is easier to basically break down, whereas the lipids requires a bit more time. And I think particularly when you're looking at high impact, high intensity exercise, lipids are not ideal for that, whereas carbohydrates are. They represent a better energy source. But if you're using maybe low intensity exercise, then lipid metabolism will probably take over. Okay. We can also use proteins as a potential energy source. Now, this I would say would be in very extreme circumstances. Your body would not want to use proteins as a source of potential energy. They would always preferentially go for carbohydrates, then lipids. If you had no reserves of either, then your body would start to literally scavenge your own muscles, your protein in there, as an energy source, as a last-ditch effort to keep you alive in starvation. So what happens there? So again, I'm going to cut to another diagram to show you how proteins are broken down to help generate ATP. Right then, so with proteins, well, the first thing that has to happen, we have to hydrolyze all those peptide bonds, so we can end up with all our amino acids. The amino acids then need to go through a process called deamination, where we remove effectively the amine group. And that usually takes place in your liver inside your body. The amine group, the amino group, however you want to word it, will end up being converted into ammonia and then urea and then excreted by the kidneys as a waste product, a way of removing excess nitrogenous waste. The remainder of the amino acids, so that will be our carboxyl group and the R group, will have a different fate depending on how many carbons are involved. Any 3 carbon bits will end up basically substituting pyruvate, so they can then be used, then they can then enter the link reaction. Anything that's 4 to 5 carbons could act as an intermediate compound within the Krebs cycle, so it can be used in that way there. So they can both feed into those aerobic aspects of respiration. So that kind of ends it quite nicely for me, and we've looked at everything we need to know in quite a bit of detail about aerobic respiration, and then next week we're going to look at anaerobic, which sometimes people find a bit tricky. I don't think it's nearly as complicated as anything we've seen yet actually, but sometimes just the thought process behind it, we maybe didn't miss it. So I'll hopefully anticipate where you might find it tricky and we'll sort it out. So this is Dr. P signing off. Stay safe, have a good one. Cheers.